Happy Monday. Thank you. No one tells me good morning. Welcome to lecture 56 of BMS 300. We are now on to part 3 of the reproductive system, and just so you know, we are going to pick up on in part 2 the female reproductive system. We're going to start with contraceptives, so if you want to just make sure you have the right slides up, just a heads up for that. Before we get started, I just want to let you know that exam 4 results will be released later today. And I was asking around for what is the average, right? And the average is 82, and with a range of 31 to 110, right? So I hope this encourages you to prepare well for the final exam. Pick up your old exams and make sure that you are familiar with the questions and the format of questions in that. Are there any questions for me before we get started? Okay. Let's begin with an overview of what we'll be covering today. So when we stopped on Friday, this was one of the last slides we looked at. We were talking about the hormonal control of the female reproductive system function, and so I told you how there are changes taking place in the ovaries as shown by the second panel here. There are also changes taking place in the uterus, and these changes are being regulated by hormones which are secreted from the hypothalamic pituitary axis as shown by the first panel, and then the third panel is showing you what are the hormones released by the ovaries. So we need to understand how this hormonal regulation works in order to appreciate how contraceptives based on hormonal regulation will function. And there are two types of contraceptives we'll talk about. You have the short acting and the long acting ones. We really won't get into the details of how barrier contraceptives work, but that is what will help to decrease the risk of sexually transmitted diseases. So we'll talk about some of the more prevalent sexually transmitted diseases, and after that we'll move on to talking about the determinants of sex. So this would be the part 3 slides which I uploaded. So when we talk about the determinants of sex, we see that there are 3 levels of differences. You have differences at the chromosomal level between males and females, and then you have differences in the gonads, and these in turn will have an impact on the phenotypic differences seen between males and females. So this is something we'll talk about, and then I'd like to introduce you to some experiments done by this scientist called Alfred Yost. So he was a reproductive endocrinologist who would conduct fetal surgeries, right? And so we'll talk briefly about the experiments he did to help us better understand how which gonads release which type of hormones for the specific sexual phenotype. And then when we come back on Wednesday, we'll talk about some of the disorders of sex development, right? So this could be disorders in terms of chromosomes, and that could lead to either Turner syndrome or the Klinefelter syndrome. Those are 2 examples we'll consider. And we'll also see that abnormalities in the way hormones function can also lead to disorders. So one condition is the androgen insensitivity syndrome. So this is a problem with the receptors for androgens, and that can lead to this condition here. And finally, a congenital adrenal hyperplasia is when you have an excessive amount of the adrenal glands producing androgens, right? So these are conditions we'll talk about when we come back on Wednesday, and it'll really make sense when you understand what are the 3 determinants of sex. So to get started, we see that there are 2 types of contraceptives. You have the short acting ones and the long acting ones, which can have their protective or their contraceptive function for several years. And so we will mainly focus on the contraceptives which are based on hormonal preparations. So in this picture here, I want to tell you that we will not be talking about things like the cervical cap, the diaphragm, or the internal condoms in females or the external condoms in males. These are just barrier methods which will prevent the sperm from fertilizing the egg, or even spermicides which will kill the sperm, right? So these are things we won't talk about. What we will focus on is things like the pill, and this is the combination oral contraceptive pills, also abbreviated as OCPs. This has a combination of both estrogen and progesterone. And then we'll talk about the contraceptive patch. This can be put on the skin, and this will release the hormones. And also the vaginal contraceptive ring is also capable of releasing these two hormones. But in order to understand how they function, we need to go back to this slide. So we see that estrogen, when you have a peak in estrogen, it's mainly going to increase the proliferation of the uterine lining. So that is one thing it does. And then progesterone, on the other hand, we see that it's going to have a negative feedback effect on the GnRH, gonadotropin releasing hormone, which will in turn decrease the which when suppressed will decrease the FSH and the LH levels. So without that, you will not be able to have follicular genesis or even the ovulation. And the purpose of these two hormones being given in the pill form is to replicate what is happening in the body. So by having a 3 weeks of exposure to estrogen and progesterone, if you notice here, it's like going to replicate what's happening in the body, and then having a 1 week free of these hormones is going to lead to the menstrual phase. Right? So that is the main goal. But let's walk through this step by step. So we find that these hormones will impair folliculogenesis. Right? Let's stop with that and consider how this takes place. So when you think about progesterone, notice that as you have high levels of progesterone, you're going to have a decrease in GnRH because it has a negative feedback effect on the release of the gonadotropin releasing hormone. So GnRH levels will drop, and if GnRH levels drop, two things happen. So what are the other two hormones which won't be produced? What are they? What are the other two? It's right here. FSH and LH, right? So FSH, if it's not being produced, then you won't have follicular genesis taking place, right? And if the follicles don't develop, then the eggs cannot be released. And also we find that if the luteinizing hormone is also not produced, we see that ovulation cannot take place because you need luteinizing hormone for ovulation, where the developed follicle will rupture and release the oocyte. So I hope this makes sense why having the progesterone is going to impair follicular genesis and ovulation. Are we clear on this point? Yes. We'll be referring back to this repeatedly. Okay. So we also find that exposure to the estrogen is going to have two effects, right? Firstly, it's going to cause the proliferative phase to occur, where the endometrial lining will start multiplying, and then you have this proliferative phase. And then once you have this increase in the progesterone level, it's going to transition from the proliferative phase to the secretory phase. So one has to follow the next, and that is how it's going to mimic what's going on in the female reproductive tract. And then when you have a removal after this exposure, it's going to induce the menses. And this removal, what it is trying to emulate is the fact that the corpus luteum will degrade. And once the corpus luteum degrades, then there's no more progesterone being released. And when progesterone isn't released, you have again GnRH levels going up as well, right? So when you have a drop in progesterone levels, that's when the menstrual phase will be triggered. Is this clear? How estrogen and progesterone, how they're going to replicate what's going on in the body, and then when there's that interval where there's no exposure to the hormones, it will lead to the menstrual cycle. And when you have the menstrual flow, you can maintain a pregnancy. Are there any questions? Okay, if there are no questions, we see that this method is rapidly reversible as dictated by the user. So if you were to stop taking these hormonal contraceptives, then pregnancy is possible. And the failure rate in this method is quite high. It's 7%, and this has got the highest failure rate because it is prone to misuse. So forgetting to take the pills in their regular schedule could lead to failure of this method. Now, while we're still on the topic of hormonal preparations, we see that some women may have contraindications for estrogen, right? So what we mean by contraindications is that they have a risk for certain conditions. For example, some with hypertension. If they take estrogen, it can further worsen that condition. Other things include, like if they have a propensity to, say, breast cancer or cancer of the uterine lining, then taking estrogen hormones can also exacerbate that risk as well. And even I told you that estrogen has got an effect on the central nervous system. So those who have concerns like neurocognitive disorders or even dementia, they can have contraindications to taking estrogen. So in such cases, they only would use progesterone in the preparation. So this is called a mini pill, or the POP, progesterone only pill, and these could be taken in the form of injections as well, the progesterone dosage. And the impact of progesterone alone is going to have an impact on the reproductive tract, right? It can also have an impact on the hypothalamic pituitary axis as well, to impair folliculogenesis, but we see progesterone alone can impact the reproductive tract. And so what happens is it can cause the thickening of the cervical mucus, and as the mucus gets thickened, it can block the sperm. And we find that the progesterone can impair the endometrial development. So I told you in the previous slide that the proliferative phase is mediated by estrogen, right? Whereas the secretory phase, the transition to that is mediated by progesterone. So by just giving only progesterone, what will happen is that the mitosis of the endometrial lining will be impaired, and if the mitosis is going to be impaired, then the proliferative phase cannot take place, and that will lead to the early secretory phase. So it's going to have an impairment in the endometrial development. Do I need to clarify how progesterone can impair the endometrial development? It's going to mainly affect the mitosis of the endometrial cells, and that will impair the development of the endometrial lining. Are there any questions at this point? Okay. We also find that progesterone can prevent ovulation to variable degrees. So when we see that 50 to 90% of the cycles can be prevented. And the reason why we say it is variable is because it really depends on the concentration of the progesterone and the size of the individual. So if the individual is small enough, then the concentration of the progesterone may be sufficient to affect the hypothalamic pituitary axis, and so that could even prevent ovulation. So if you have enough if you have enough progesterone which is blocking GnRH release, that can affect the release of the luteinizing hormone, so it can prevent ovulation. Is this clear how the progesterone only pills work by preventing ovulation in some cases, but mostly it's going to affect the reproductive tract. So this too is rapidly reversible, and it's dictated by the user. So you stop taking the pills, over time the person can be reproductively fertile again. So the typical failure rate is between 4 to 9%, and again here the reason is it is prone to misuse. So forgetting to take the pills can lead to the failure of this contraceptive. And we find that the low dose preparations of progesterone can lead to unpredictable bleeding, because we see that it can have an impact on the menstrual cycle depending on the size of the individual. So in some cases it can lead to unpredictable bleeding as well. Are there any questions at all, folks, on how these hormone preparations prevent pregnancies? Okay, moving on, let's talk about the longer acting reversible contraceptives. So this is also abbreviated as LARC, and this can provide contraception for up to 12 years, right? So some cases it's fewer than 12 years, and let's consider what they are. So this would include the insertion of an intrauterine device, as shown here, like this copper IUD coil, or subcutaneous implants which can be implanted. And we find that these approaches for longer acting reversible contraceptives, they are going to require access to a healthcare professional. So it's not something the individual can implement on their own. And we find that the hormonal IUDs, they are capable of releasing progesterone into the uterus for up to 7 years. And again, progesterone will have an impact on the reproductive tract. It's going to thicken the cervical mucus, which can block the sperm. It can also cause the thinning of the uterine lining. I told you how progestin will have an impact on the endometrial development, right? And so it can cause the uterine lining to thin, and in some cases it may prevent ovulation if the concentration is enough to trigger the hypothalamic pituitary axis. We also have copper IUDs, and these can provide a mechanical barrier up to 12 years. And what these copper IUDs do is they're going to block the sperm from reaching and fertilizing the egg. So they will make the environment very hostile for the sperm, so the sperm will die. And even if in some cases the fertilization does happen, the presence of the copper IUD will prevent implantation from taking place. But the main thing to remember is that the copper IUD does not inhibit ovulation, because it's not having an impact at the hypothalamic pituitary axis, unlike the hormones, right? So this does not inhibit ovulation. There are also hormonal implants, which are these matchstick sized flexible rods, which can be implanted under the skin, and it will release progestin up to 3 years. And so I didn't mention this, but progestin is just the synthetic form of progesterone. And the mechanism by which these hormonal implants work is similar. They are going to have an impact locally to prevent the sperm from fertilizing the egg by thickening the mucus, or by making the uterine lining thin so that implantation can take place, and in some cases it can also prevent ovulation. Now the typical failure rate of the long-acting contraceptives is a lot lower, mainly because it is put in place by a professional, right? Whereas the short-acting ones, they are prone to user misuse, and that's the reason why they have higher failure rates. So moving on to emergency contraception, we see that intrauterine devices can be used in cases up to 5 days after unprotected sex, and this can reduce the likelihood of pregnancy by more than 99%, and this is said to be the most effective form of contraception in the case of an emergency. There are also cases of morning after pills, and I want to tell you that there are a couple of these, right? So it can be taken up to within the 5-day window of unprotected sex, and it can reduce the pregnancy likelihood by 75 to 90%, right? But we find that it is most effective if it's taken within the first 3 days after unprotected sex. Now there are two types of these morning after pills. One is the levonorgestrel, also abbreviated as LNG or ulipistral acetate. And the difference between these two is that it should be taken based on the body weight of the person. So LNG is a synthetic progestin, and this will block the LH surge. And you know how it blocks the LH surge, because progesterone will impair the release of GnRH. If GnRH isn't released, then the luteinizing hormone won't be produced as well, right? And it can cause a delay in ovulation. And this will be less effective in those who are over 155 pounds. So it's more helpful for those who are lighter than that weight. However, for those who are heavier, we see that ulipistral acetate is a progesterone receptor modulator. So this will go act on the progesterone receptors, and it will similarly block the surge of the luteinizing hormone, and also it can prevent ovulation. You need the luteinizing hormone for the follicle to rupture and to release the egg. And we know that the luteinizing hormone surge will be impaired if it has a negative feedback effect on GnRH release. So this has a greater efficacy in those who are over 155 pounds, but it is less effective in those who may be over 195 pounds in weight. So we see that the most effective way of preventing pregnancies is also the least popular. And what is that? What is the most effective but least popular way? Abstinence, right? So very good. So we see that sexually transmitted diseases or sexually transmitted infections, these terms can be used interchangeably, and we said anyone is at a risk for these STIs or STDs, right? Anyone having oral, anal or vaginal sex is at a risk. And so what I'm going to do now is I'm just going to give you a few examples of these sexually transmitted diseases, and I will mention the name of the disease and I'll tell you what is causing it, right? So that's all I'll hold you responsible for. You need to know whether a given disease is caused by a bacteria, virus or a protozoan, right? That's all I'll hold you responsible for, and I'll explain why I give these examples later on in today's lecture. So the first one is trichomoniasis, and this is caused by a protozoan parasite, right? And the name of this is Trichomonas vaginalis, and this can be cured with antibiotic treatment. The next one is syphilis. This is a bacterial infection caused by Treponema pallidum. This too can be treated with antibiotics, but if the antibiotics are not given in a timely manner, it can affect a number of organ systems, and at that point it's too late, right? What damage to these organ systems cannot be reversible, so it's important to intervene quickly in the case of syphilis. Then you have general HPV. This stands for HPV stands for the human papilloma virus. This is caused by a virus, and there is no cure for this, right? So once infected, there is no cure, but there are ways to treat the symptoms, like if there are genital warts that can be removed, but once infected with HPV, there is no cure. Next example is gonorrhea. This is a bacterial disease caused by Neisseria gonorrhoeae, and this can also be treated with antibiotics, but because of the increased use of antibiotics and the misuse of antibiotics, more resistant strains are developing, right? So that is something to consider. And then there's chlamydia. This is a bacterial infection caused by Chlamydia trachomatis, and this can be treated with antibiotics. And finally, probably the most commonly known one is HIV, the human immunodeficiency virus, which if left untreated can lead to the acquired immunodeficiency syndrome, AIDS. Okay, so these are some of the examples of sexually transmitted diseases, and all I would hold you responsible for is know what kind of pathogen causes it. Is it a protozoan, virus or bacteria? Sounds good? Okay, pretty straightforward. Okay, so now I'm going to give you a few statistics, and if you look at your handouts, you'll know that it's not something I'll test you on, but just know that you'll have a general idea of which is the age group which is at a highest risk for these diseases. So this is data based on CDC's website. So CDC is the Center for Disease Control. So they found that in 2021, 50% of more than half the new cases of HIV were individuals between the age of 13 to 34. Right? So it is those who are younger where there was more than half of these diagnoses done. And then if you look at the differences in terms of males and females, we find that in both males and females, it's the age group of 25 to 34 which seems to have the highest HIV diagnosis, right, in the US. And if you look at the case of chlamydia, again it's between the ages of 15 to 24 where you have the highest number of reported cases. And notice that these are 800 to close to 2,000 cases per 100,000. That is how they report this. The numbers may seem like a small percentage, but notice that the age group which is most affected is between the ages of 15 to 24. So in the case of gonorrhea also, both in males and females, it's the age group of 20 to 24 who are most affected by gonorrhea. And similarly in syphilis too, you have the age group of 20 to 29. So they're all young adults, right, who are mostly affected by these sexually transmitted diseases. So why did I mention these diseases? Well, the reason is that the most common STDs in the college age people include chlamydia, gonorrhea, syphilis and the human papilloma virus. So that is the reason why I mentioned these examples of sexually transmitted diseases. Now the interesting thing is that the people who are at the peak of their health in their life is this age group, right? But the question is why are they the most susceptible? And the reason is that this age group is also associated with a lot of risky behavior, right? And this includes binge drinking. So when you drink too much, your judgment is impaired, leading to unprotected sex, the opportunity for multiple sex partners, and also unintended pregnancies can also result from the risky behaviors. And in recent times, we find that it is the rise in the use of dating apps, right, which seems to facilitate this hookup culture, right? And one of the biggest lures in these dating apps is the sense of anonymity, right? And this makes it very difficult for public health officials to carry out contact tracing. So back in the day when it was not as anonymous, if one person was infected, they could find out how the infection was spreading. But with these dating apps, it makes contact tracing very challenging. So there is another age group where there seems to be an increasing risk in sexually transmitted infections, and this is in the elderly, right? So people in their late 60s and onward, right? And the reason for this is there are a few reasons causing this. So there's increasing use of Viagra, which can treat erectile dysfunction, and as a result, even older men can be sexually active. And also there's a lack of condom use, because when you're at that age, you're not thinking about the risk of pregnancies. And also there is an increase in the midlife crisis, like divorce rates increasing or the death of a life partner. And this age group seems to be less inclined to talk to a health provider about sexually related diseases, and there's less screening as a result. Right, so that's all I have to say about sexually transmitted diseases. And what can potentially decrease the risk is barrier methods as contraceptives, which can decrease the risk of sexually transmitted diseases. Any questions, comments? Okay, so let's move on to sex determination, right? So we see that there are 3 levels of differences between males and females as far as sex determination is concerned. The first one is the genetic differences. Secondly, you have differences in the gonads, so gonadal differences, and also phenotypic differences. And let me walk you through each of these, right? So what we mean by differences in chromosomes is that in males you would have an X and a Y chromosome. In females you would have 2 X chromosomes. And as far as the gonads are concerned, in males you would find testes, and in females ovaries. And the phenotypic differences include differences in the reproductive tracts, right? So in males, this starts with in the testicles you have the epididymis, which leads to the vas deferens or ductus deferens, that leads to the ejaculatory duct. Then we talked about the 3 different accessory glands, bulbourethral vesicle, the seminal vesicles, and the prostate gland. And the external genitalia is the penis and the scrotum. Whereas in females, we talked about the oviducts leading to the uterus and the vagina. The accessory glands, we only talked about one, right? We talked about the greater vestibular gland in females, and also the mammary glands. And external genitalia includes the labia minora, majora, and also the clitoris in females. So we'll go through each of these differences in a little more detail. So when you think about the differences in chromosomes, we find that every cell has got 23 pairs of chromosomes, right? One from mom and one from dad. So in those 23 pairs, the first 22 pairs are called autosomes because they carry traits for the general characteristics, right? So those 22 pairs of chromosomes, they will have the genes for general body characteristics. Whereas the last pair, the 23rd pair shown right here, you could have either an X and a Y chromosome or 2 X chromosomes, right, in females. So now let's see what is the difference between the X and the Y chromosome. So we see that the X chromosome, as you can see in this picture here, is much larger than the Y chromosome, and as a result it makes sense that the X chromosome has got 900 genes, right? It's got a lot of genes. The Y chromosome will have a lot fewer. And we see that one functional X chromosome is necessary for survival. So if there was a fetus with no X chromosomes, or maybe just a Y chromosome and no X chromosome, that would be fatal, right? So X chromosome is necessary for survival. Now the Y chromosome, on the other hand, it's much smaller. It has only 55 genes, and there are a few things which are important, but we are only going to consider one of these genes, right? And so the gene we are interested in is the SRY gene. So this stands for the sex determining region Y, right? So the sex determining region Y gene, right? And so it is this gene which is going to start the development of the male characteristics. And there was a study done where they took a female mouse fetus and they expressed the SRY gene in it, and the mouse was able to develop testes, but then it could not produce sperm because there are other things in the Y chromosome as well, right? So we see that SRY is necessary for the testes production. And you find that the Y chromosome is not necessary for survival, right? For example, if you had if an individual had 2 Y chromosomes, that individual may be taller than average, and also there could be delayed language development. So we see that having an abnormalities in the number of Y chromosomes, having too many or too little, can lead to abnormalities. And we'll talk about what happens when there's an XO individual when we come back on Wednesday. So the question is, how do chromosomes influence the gonadal development? So how can the chromosomes influence whether it's a testes or ovaries developing? So we see that the gonadal development is only dependent on the genes. Hormones do not affect gonadal development at all, right? So if you were to take a female mouse and inject it with testosterone, it's not going to develop testicles, right? It has to be based on the chromosomes. So now let's look at the inheritance of the X and the Y chromosomes. So we see that the female parent will have the eggs. Each egg is haploid, meaning it's only got one copy of the genetic material. The male gamete, the sperm, is also haploid. It's only got one copy of the genetic material. And we see that if a X sperm fertilizes an X egg, it will lead to a female offspring. On the other hand, if a Y sperm were to fertilize egg, which is also X, it will lead to a male offspring. So it is the sperm which determines the sex of the offspring, right? It's not the egg which determines that. So if there's at least one Y chromosome, the testes will develop, right? And if there is no Y chromosomes, there will be the development of ovaries. So now let's see what is happening in order for the Y chromosome to influence the testes development. So we see that until the 6th week of development, the gonad is said to be bipotential, right? It can either go this way or the other way, right? It can become a testes or an ovary. So we see that when you have the Y chromosome, you will have the SRY gene on the Y chromosome producing the SRY transcription factor. Okay, so the SRY is the transcription factor produced by the SRY gene. So this SRY transcription factor is going to cause the upregulation of SOX9, which is another transcription factor. So SRY will lead to the upregulation of SOX9, another transcription factor. SOX9, on the other hand, it will inhibit the expression of RSPO1, beta-catenin, and Wnt4, right? So beta-catenin is a transcription factor, whereas RSPO1 and Wnt4, they're all genetic factors, right? And because these three are being inhibited, we find that the testes will develop. And so we see that, just to repeat what I said in words in text, we see that the SRY upregulates SOX9, which will block RSPO1, beta-catenin, and Wnt4. And that's the reason why the testes will develop. Now the important thing to keep in mind is that RSPO1, beta-catenin, and Wnt4. So all 4 of these, they are autosomal factors present in every single cell, right? And without the SRY transcription factor activating SOX9, we will find that the ovaries would develop, right? And so notice what happens here. If you have 2 XX chromosomes, RSPO1, beta-catenin, Wnt4, all those 3, they will block SOX9. And once SOX9 is blocked, we'll see that the ovaries will develop as default. Are there any questions? So we see that the default development is the ovaries, but in order for the testes to develop, these 3 factors need to be blocked, and that can be blocked by SOX9, which is also an autosomal factor, but that has to be activated by SRY, which is found only on the Y chromosome. Any questions at all, folks? So what I've shown you here is a very simplified perspective of how this happens, and this level of detail is sufficient for our level of discussion. Specifically for understanding the disorders of sex development, this level of detail is sufficient. So any of you who are curious to know how things happen, this is a more complicated view with extra details. I've not given you this slide in your handout because it's way beyond the scope of what we are discussing, right? But I just hope you know that I've given you a very simplified perspective. Okay, so now that we understand how the testes and the ovaries develop, we need to understand how did the internal ducts develop, right? So we see that before sexual differentiation, an embryo is going to possess both male and female reproductive tract progenitors. So it's got these progenitors which will turn into male or female reproductive tract, but both are present in the embryo as it is developing. So we see that the Mullerian duct is the female reproductive tract progenitor, and the Wolffian duct is the male reproductive tract progenitor, and I hope this color coding helps you to know which ones they are. So we see that the Mullerian duct, it's going to remain in the female as the female develops, whereas the Wolffian duct would remain in the male. And so this Mullerian duct is going to differentiate into the oviducts, also known as the fallopian tubes, into the uterus, the cervix, and the upper part of the vagina. So you remember I told you how the uterus and the vagina are part of the ducts, even though it doesn't look like that, but here's where we get that information from. It is from this Mullerian duct where the oviduct, uterus, and the cervix and the upper part of the vagina will develop. Now on the other hand, it's the Wolffian duct which will develop into the epididymis, which will carry the sperm from the testes, and it will also develop into the vas deferens and the seminal vesicles as well. So the seminal vesicles is an accessory gland, but all of this will develop from the Wolffian duct. Are there any questions? Which is the male and female progenitor of the tracts? Is this clear? Okay, so now we will talk about the experiments done by Alfred Yost. So he was a reproductive endocrinologist with super steady hands, right? So what he did was he did surgeries in the fetus. So the rabbit fetuses, he did surgeries to take out the gonads. He would either remove the testes or the ovaries from these fetuses and put them back into the reproductive tract and let them develop, right? So those were the experiments he did. So one of the major findings is that when both whether testes or ovaries were removed, the adult female rabbit would develop, right? So regardless of whether ovaries or testes were removed, the female rabbits would develop. And so we see that regardless of the genetic sex, regardless of whether the fetus was XX or XY, it acquired a female phenotype, right? So what we mean by this female phenotype is that the Wolffian duct would regress, whereas the Mullerian ducts were maintained. So in order for this female phenotype, the Wolffian duct would regress, but the Mullerian duct would remain. So this was only the first set of experiments, but he did 3 sets of experiments, right? So the first experiment, as I mentioned, the gonads were removed. The XX fetus had the Mullerian duct, and so did the XY fetus as well when the testes were removed. So what does this mean? So what this means is that neither the testes nor the ovaries are required to maintain the Mullerian ducts. So the female progenitor tracts, you don't need anything to maintain it, regardless of whether you take out the testes or the ovaries, the Mullerian duct will be in place. So in the second experiment, what he did, he took out the gonads, but he decided to add a testosterone crystal, right? He put in a crystal of testosterone, and what he found was that both the Mullerian and the Wolffian ducts remained. Okay, so how do you interpret this, right? So we know that the presence of the Mullerian duct doesn't need anything, but we see that the Wolffian ducts remained because of testosterone. So testosterone was required to maintain the Wolffian ducts. Are we clear, folks? Testosterone was required to maintain the Wolffian ducts. Okay, now let's move on to, can we move on to experiment 3? Okay, so experiment 3, he removed the gonads, but he added a small fragment of testes, right? So we'll consider the experiment only in the XX fetus, because it doesn't make sense to take out the testes from a XY fetus and put the testes back in, right? So that's just plain mean. So what happened was, when in a XX fetus, when the ovaries were removed, right, but the testes fragment was put in there, it resulted in only the Wolffian ducts remaining. The Mullerian duct disappeared. So we know that the testes will produce testosterone. Yes, do you remember what cells produce testosterone? Leydig cells, also known as the interstitial endocrine cells, right? So we know that the testes fragment was producing testosterone to maintain the Wolffian duct, but we also see that the testes was releasing something else, right, to cause the regression of the Mullerian duct, and this something else was appropriately named as the anti-Mullerian hormone, abbreviated as AMH. So do you remember what was the cell which produced AMH in the testes? We only talked about 2 kinds of cells, also known as nerve cells. So I told you the nerve cells. These were the really massive cells, right, which extend all the way from the basement membrane to the lumen, right? So this is just asking you which cells in the testes produce testosterone and AMH, and I hope you know these names. So is this clear how he figured out, using these experiments, what was determining whether you have the Wolffian duct or the Mullerian ducts developing? Pretty clever experiments. Okay, so you have a chance to copy down. Can we move on? Yes. Okay, so we see that this is just to review what we just discussed. So we see that before differentiation, there's both the Wolffian and the Mullerian duct shown in this first panel, and then to your left hand side we see how the differentiation to males takes place, right? So we see that the testes will produce the anti-Mullerian hormone, which causes the regression of the Mullerian duct, and testosterone will lead to the maintenance of the Wolffian duct. So the way I would help you to remember it is that the wolf must be fed with testosterone lest it perish. Okay, so I hope this helps you keep the details in your head. So anti-Mullerian duct will destroy the anti-Mullerian hormone will cause the regression of the Mullerian duct, whereas the testosterone is necessary for the Wolffian duct to remain. So now we've just talked about the internal reproductive tracts. What about the external reproductive structures? So we see that the phenotype consists not only of the internal genitalia, which are the reproductive tracts which I mentioned, but also the external genitalia is also formed by the hormones, right? And we see that in the ovaries they'll produce estrogen and progesterone will talk about its function in maintaining the pregnancy, right, when we come back for the next lecture. And we also talked about progesterone in preventing pregnancies. But it is estrogen which mainly plays a role in the development of the breasts and the uterus and also the vagina and the ovaries. So the initial maturation of these various structures in the female reproductive system is possible through the action of estrogen. Okay, so that is why I want to highlight in this slide. Now, what about males? What do the hormones from the testes do in the development of the male phenotype? So I told you that the anti-Müllerian hormone will get rid of the Müllerian duct, whereas testosterone, it can be converted into DHT, also known as dihydrotestosterone, by this enzyme known as 5-alpha reductase. The good news is I will not hold you responsible for these structures, but I do hope you know what DHT does. So dihydrotestosterone is going to make up 10% of the circulating levels of testosterone, and this is also called as the super testosterone because it is more potent. It can bind to the same receptors more strongly. And we find that there is also estradiol, which is the most potent form of estrogen in males as well, and it's the most potent form of estrogen, and 70% of the estradiol is going to be produced by the conversion of testosterone into estradiol, and the enzyme involved in this is aromatase. And we find that estradiol is found in small amounts, 2 nanograms per deciliter, and you may be wondering, what is estradiol doing in males, right? It's actually pretty important. It's necessary for the sex drive, it's necessary for spermatogenesis, and also for maintaining an erection. Estradiol is important. So if you don't have estrogen produced in males, that male could be impotent. So we find that by week 6, the external genitalia is still undifferentiated, right? So here is a cloacal opening, and if you notice on either side of the cloacal opening you have this genital swelling, and the genital swelling will eventually turn into the labia majora and the minora, right? And we find that feminization is the default pathway taken. And also this genital tubercle shown here is what will differentiate into the clitoris in females. And we find that all of these external genitalia will be formed by default in females, right? Whereas for in males you need dihydrotestosterone for the masculinization, where the genital tubercle will turn into the glans of the glans penis, and then the genital swelling is going to form the scrotum, right? So dihydrotestosterone is necessary for the masculinization of the external genitalia. So now we've seen how the 3 levels of sex determinants work: the role of the gonads, the role of the hormones produced by the gonads, and also the role of the chromosomes. So let me just summarize what we've discussed. We talked quite a bit about the hormonal regulation of the female reproductive function through the hypothalamic pituitary gonadal axis, and we saw how this explains the functioning of hormonal contraceptives, both the short-acting and the long-acting ones. And we see that there are different types of STDs, and the risk can be minimized with barrier methods, but the most effective is the least popular, which is abstinence. And we saw how there are different populations at risk, including both the elderly and the young people as well. And we see that the chromosomes will influence the development of ovaries or testes, and the Alfred-Yost experiments reveal the role of hormones in the development of the internal and the external genitalia. And when we come back on Wednesday, we'll talk about some of the disorders of sex determinants, where there could be problems in the chromosomes or in the levels of the hormones and the receptors for those hormones as well. Thank you all for your time, and I will see you on Wednesday.